Now that we've taken a look at the nervous system and how it's put together, we can start to take a look at how we're going to get to that transmission of signals from one neuron to the next cell. One of the things we have to take a look at is how we're going to move ions in between the cells themselves. And so we'll take a quick little look here at the different proteins that make up the ability to move substances in and out of the cells. And so we've seen this before when we looked at the cells. And now we'll look again at some of those proteins that are more specific to the nervous system. So part of what we have going on is that we've created a situation where we have essentially an excess amount of sodium on the outside of the cells and an excess amount of potassium on the inside of the cells. And just the normal concentration gradient wants to have those potassium ions move to the outside of the cell and the sodium ions to move to the inside of cells. And so this can happen just simply by using what we call leak channels. And so these are always open. They're available for each of the ions to move through, just simply moving down their concentration gradients. There's no energy required. We don't have to force the process itself at all. It's just going to simply move through those constantly open leak channels to try and balance out the membrane. We also have mechanically gated channels, and this we can use on certain neurons when we want to have them open up based upon some sort of applied pressure that is happening in and around that particular channel that's there. So they're mechanically gated, which means that if we deform them in some way, we can utilize this for things like touch. We can utilize this in baroreceptors for determining things like pressure. But either way, it's kind of, in a sense, if you think about kind of like a beach ball, and on that beach ball you have like the little valve that's there. And in order to get that beach ball to deflate or even to inflate, we've got to push on that little valve and deform that valve to get it to open up. And that's going to allow our air to come out of that beach ball. In this case, it's going to allow our sodium ions to move into the inside of the cell, and we're going to get depolarization of the cell. We're going to get it to where the cell is now starting to depolarize, and we can have some sort of action potential or a graded potential. Chemically gated, this is where we'll see the most of our channels in this way. We have a particular protein channel here that has a binding site for some sort of neurotransmitter. In this particular case, it's acetylcholine. So acetylcholine is going to be our substance utilized here. And when acetylcholine is absent from that binding site, our gate remains closed. And so our chemically gated channel is closed when there's no acetylcholine there. But as soon as we put that acetylcholine into the receptor site, that receptor site causes a change in the shape of our receptor, of our gate here. Our gate opens up, and now sodium ions can come to the inside of the cell. And so they're again going to want to be going down their concentration gradient. We've got excess amounts of sodium on this side. We've got a lack of sodium on this side. As soon as that acetylcholine gets removed from the binding site there, it then recloses up our gate. And so it's going to close once again, and we're no longer going to be able to have that sodium rushing into the inside of the cell. And so acetylcholine has to be broken down. We have to break down that particular chemical there. And in the case of acetylcholine, we'll use acetylcholinesterase. Some other channels are voltage gated, and so they're going to be kind of like an electric door. So you go into a store or something like that, and there's a door that's there that when we apply electricity to it, it opens up. Our voltage gated channel is kind of the same way. And so when we have a situation where the outside of the cell is more positive and the inside of the cell is more negative, we'll talk about that, this potential that's there in an upcoming talk, and how we get it to be like that. But when we have that situation, our gate remains closed. And so it's going to stay closed at that period of time. If we can reverse the voltage, though, if we can make the inside here more positive, that's going to cause this activation gate to open up. And so that activation gate then causes the the cell to start to allow sodium once again to come into the inside of the cell. It's again following its concentration gradient. We've got excess amounts of sodium on the outside of the cell compared to the inside of the cell, and it simply follows that concentration gradient. Once that charge starts to change, and so once that charge is no longer a positive on the inside, or sometimes it's a timing issue, we get the inactivation gate closing. So this is going to close up our gate, and it's no longer going to allow sodium to come into the inside of the cell. And so we once again are, in a sense, at a stage where now the inside of the cell reverts back to our resting state, and that movement of sodium ions is no longer going on. Lastly, we have a sodium-potassium pump. And so sodium-potassium pumps are there for specifically sodium and potassium. And so potassium is there, just circled the phosphate groups there. So this is our ATP, our adenosine triphosphate. So we have 3 phosphate groups on there. Over here we have our potassium. And it's going to be a pump to where always we get 3 sodium out and 2 potassium in. And so it's always going to have this situation where we're pumping sodium out and potassium in a ratio of 3 to 2. And so 3 sodiums out, 2 potassium in. It does require energy here, so we do need ATP in order to have this occur. So we're going to break that ATP down into ADP, which is our adenosine diphosphate, and we're left with an extra phosphate group there. That phosphate group is going to leave the binding site there, and when it does so, it changes the shape once again of that potassium, and now that potassium can come into the inside of the cell. So it's kind of like a 2-pronged process. When ATP comes in along with our sodium, it allows for, and it becomes hydrolyzed, that phosphate group that is left there opens up, sodium can leave the inside of the cell. Potassium now can come in, and it fits into those triangular structures that are there. It comes into the inside of our receptor here, and then once that phosphate group leaves its binding site, it then allows the gate to open up, and it comes to the inside of the cell. And this is going to be the thing that creates this dynamic where we have excess amounts of sodium on the outside of the cell and a lack of potassium on the outside of the cell. So the potassium is going to be all inside the cell, and the sodium is going to be out. But again, we go back to those leak channels, and the sodium is going to slowly leak back in. Potassium is going to leak back out a little bit faster.